We are delighted to have Professor Annapurna Purduri from Harvard University is here with us, another great friend of the community. And Anne does many things. You're on the board now of AES, aren't you? At Board of Directors of the American Epilepsy Society. She has her own lab at Harvard University where she does translational research, and she is an expert in genetic epilepsies, particularly in KCNQ2. She's been working for some years now on a natural history study for us at KCNQ2, and not just here at the actual foundation, but just broadly within the KCNQ2 DEE community. It is my real pleasure to bring to the podium Anne Purduri.
Thanks so much, Sarah, and I'm really pleased and humbled to be here with all of you. It's always great to see people in person, but after this long hiatus, it's even nicer, so thank you for making it happen to all of you. I just have to wake up my computer here. There we go. All right. Well, to riff a little bit off of Sarah's introduction earlier about Willem de Vleming, how the improbable has such impact, I want to start by saying how much the rare has such an impact on the common. So, we're in a lot of Epilepsy Foundation, American Epilepsy Society, a lot of meetings where we're the rare, we meaning KCNQ2, or the rare genetic epilepsies, and the broader academic community and physicians often come and say, well, why does this matter? Well, first of all, it matters because your children have these conditions and children around the world have these conditions, so that doesn't need any justification. But it also matters because what we're learning from KCNQ2, what we're learning from the rare, will continue to inform the common epilepsies, which are very common. So I just wanted to start with that because it's really quite powerful, and it's what motivates a lot of the epileptologists in our group and around the country to pay attention to what's going on with the rares, because it really does have a broader impact. So I'm going to do a little bit of reminiscing about the past, but then talk about the present and the future in terms of what we're learning and some ongoing efforts in our group and in many collaborations with others. And I'll just hint a little bit about precision trials and trial readiness. And I'm going to try to remember, but as a little memory stick for myself, I put little purple stars to remind myself to just mention as we go along, what is it about what we're doing now that's going to inform our ability to give our partners in industry the insight on the outcomes that our patients and families are looking for. What do we think are meaningful outcomes for the trials that you're all coming up with? So hopefully that'll come through. So a little bit of background on the KCNQ2 natural history study. This goes back many years, as has been mentioned. We've had a number of chances to meet, and I'm putting it in quotes because natural history really, to the purists, will imply a longitudinal perspective assessment of kids where we'd see them and do assessments, history, examination, EEG, and so on over time. And we haven't been able to pull that off yet. But what we're hoping to do through a registry is to really get parts of that story told enough so that we really can be ready for the clinical trials that are emerging. To zoom out a little bit from just KCNQ2 across all of the rares, I just wanted to quote our friend and colleague Hilary Savoy from the Cute Syndrome Foundation, who provided this quote for a meeting a few months ago, that natural history studies remain elusive for most of the ultra-rare genetic epilepsies. There's rarely time or funding to lift them off the ground, and the patients and families are so exhausted from participating in the medical care that's required to just sustain their daily lives, let alone opt in for additional testing. The many retrospective and prospective data could add up to a natural history study of a kind if you know how and where to ask the right questions. So I just wanted to point out a couple of things, because when this group, the KCNQ2 Cure Alliance, got together and said, what are the barriers and how can we break them down? Some practical things came up. We needed people with talent and time, and we needed funding for those people. And when we said, well, this is how we could do this, you came through and you let us launch a registry that really has been a collection and a connecting point for a lot of different studies, to do that based in Boston, but really with a lot of different partners around the country and the world, and allowed us to ask the right questions, which were mostly your questions. So this is really very much stemming from discussions we've had at summits like this. So I hope there are more questions raised and more future studies that come from this one. So are we heading in the right direction towards our readiness? I think we are. This is from Al and Sarah's elements that Al had mentioned earlier. This beautiful timeline illustrates our knowledge about the epilepsy syndromes you've heard from John and others about, and Ingrid about the benign description now called self-knee, the eventual discovery of the potassium channel subunits that were responsible for the more benign familial condition. Later on, understanding, and nicely summarized by Sarah Vekhusen, the DEE, the developmental and epileptic encephalopathy, with refinements and expansion of the phenotype by John and others. So this timeline of what we understood in terms of the phenomenology of the epilepsy and the relationship to potassium channels, and then what we might want to do about that therapeutically, has really evolved over many decades. You know, where does this natural history study and registry fall in? For us, it really goes back to the 2017 Peabody Summit, where we met in Peabody, Massachusetts, and we had an opportunity not only to enroll families and your children into a registry, but also to enroll some of you into a translational neuroscience center study where we were able to take blood samples through a lot of very patient hours spent by you all waiting for the Beth Shidley taxi service across the way, and various other ways that we stormed the Peabody Children's Satellite. But that really launched a lot of some of the downstream studies, and so I just wanted to give a shout out to you for putting up with that. It was not exactly the easiest thing. It was we're trying a slightly modified version today with Brandon, with just the enrollment here and the collection later. But everything I'm going to mention now is really stemming from what we started there. So we've been putting together over many years now a report of the registry that Brandon's leading. We're calling it One Gene, Many Presentations, New Questions. And Brandon submitted this for publication, and part of what he's done is to call out some quotes from many of you about why did you want to take the time, even though it's a burden on you to do this, to enroll. So here's just a couple of quotes. One family said they wanted to help gain a better understanding of KCNQ2, how the gene functions, and to better understand how it impacts those with different variants. I think you're seeing evidence of that happening through Al's work and others. Because my son is now 21, and back when he was born, they didn't even have any of this knowledge. We're moving forward. We have to get more information to learn about the different genetic syndromes. And along the same lines, another parent said, my son was diagnosed with KCNQ2 quite late. He was 27. We really wanted to help the future families. So I think this really sums up the altruism, the forward-thinkingness, and really the quest for knowledge to try to get a better handle on this condition, just from a few of you. So what are we learning? What are some of these ongoing efforts? I'm pleased to say that I've had to completely redo the slides, and not a single slide from 2017 was useful today. I'm really excited about that. It did take a little bit of work, but it's, I think, amazing that we've enrolled now overall 76 families just in this registry. And I'm hoping we can kind of connect these various registries, as we talked about it during the break, with Citizen, with Rike, and some of these other endeavors, so you all don't have to keep re-entering the same information. But out of the 76 family, we had what we are considering complete data from 43, which is not to say that the other 33 pieces of data were not important, but for complete data sets, you'll see why sometimes the denominators has been important. We put this set of variants on the two-dimensional, what we call a Prater diagram of the KCNQ2 cartoon to show a few things. One is that many, many different parts of the protein are affected. These little areas here represent these transmembrane domains that Al had mentioned. There's sort of clusters of variants in some of these regions. And the dark blue circles represent the areas where you've had multiple variants, so these recurrent variants. So we had 4 with the glycine to arginine substitution at 281. I believe you had 9 in the Citizen presentation, 3 each in each of these sites. And this is important not just for sort of the epidemiology of KCNQ2 in terms of where are they recurrent, but I think as some of our colleagues are planning studies, so we have Aubrey Susi from Tim Yu's lab here, as people are thinking, okay, if I'm going to do a proof of principle study with iPS cells, or a proof of principle demonstration in a dish that we can actually treat the molecular defect that's going on with KCNQ2 encephalopathy, if they start with any of the variants, it would be useful. But if they start with some of the recurrent variants, it might be an opportunity to have replication, to have scientific rigor, really back up some of the findings. And so this is tremendously important, not just to understand the landscape, but to really drive and guide the science, especially at an early stage. I'm not going to go through all of these, but I wanted to give you a sense of how the many pieces of data that you've all been providing kind of come into a synopsis. We look at each of the variants, and as our colleagues from CHOP have been doing, we go over those variants very carefully to sort of say, well, do we believe this or not? So these are the positives. These are the likely pathogenic or pathogenic variants. There's a whole algorithm to determine whether that report that you get says pathogenic, likely pathogenic, or something else. So we've redone that. Sonal Mahida in our group has gone through and looked at all of these. We look at the seizures as a presenting onset. I'm an epileptologist like John, so we look at that as sort of the often initial feature. Excuse me, so the age of onset, what type of seizure syndrome was involved, and then other features such as developmental delay, intellectual disability, presence or absence of a movement disorder, and so on. So I'm not going to go through all of these, but I did want to tell you we really are looking at every aspect, and every bit of those surveys that you're filling out is actually being looked at and used. To summarize, we've seen the whole gamut of seizures, so everything from tonic in the early days, tonic-clonic, myoclonic, spasms, absences reported in some children, focal, atonic, spasms outside the infancy period. And they fall into many of these different syndromes that you've heard about. I don't think we need to go through all of the different names. You've heard them a couple of times. But I think that the new guidelines and the new terminology gives us an opportunity to kind of go back and say, okay, we had all of these children classified. Let's go back and let's make sure we're up with Ingrid Scheffer and colleagues' new guidelines, because we want to be up on that. It's another opportunity for data curation, another opportunity for data review to make sure that we're really appropriately characterizing these various syndromes. And this is one of the features, if we looked at all of the kids who are here who are having seizures, that would be important in terms of clinical trial readiness. You also have all of your children who are not having seizures anymore, and that's missed by this part of it. So that's really important. We also want to think about, well, what about in the next decade if we could get to those kids earlier? What if we can have a trial that would address neonates? And probably this side of the room is sort of bristling at the, oh my goodness, she wants a trial for 2-week-old babies. But yeah, we want a trial for 2-week-old babies. And so we'll get comfort with the older children, but eventually we do want to nip this KCNQ2 encephalopathy in the bud as soon as possible. And so I think this will inform us, and hopefully other features will as well. So we also saw reports of developmental delay in almost all of the kids in at least one area. This is work that Brandon has looked through, and also work we looked at with Ann Berg. A couple of individuals reported that their children did not have developmental delay, which is also important. We can't be absolute about our predictions, but we can be pretty certain when it's this evident that developmental delay and intellectual disability are important parts of this condition. We had 43 individuals asked about movement disorders. 6 out of the 43 reported a movement disorder. 35 said they did not have a movement disorder, and the others didn't respond. But the denominator here is important for a couple of reasons. We have a lot of anecdotes in clinical medicine. We say, one report is a case, 2 reports is a series, and 3 reports is this is how it is. And so, that's being a little bit flip, but that's sort of how things are as you get things started. We really would like to be more rigorous. We'd really like to say, well, there's 6 kids with a movement disorder with KCNQ2 in this group out of 43, so we can really make the point that it's there, it's important, but you're not going to see it in everybody. It might not be a feature that you'll target as a primary outcome of a trial, for example, but maybe it could be a secondary outcome that you'd look for, and maybe we should at least ask about it. And when I say we, not just in our research, but also in clinic, when you're 45 minutes into a 30-minute visit and you're talking about meds for seizures and behavior, maybe you don't get to ask about the sleep and the movement disorders and the GI symptoms and everything else, and maybe not every family is going to advocate as hard to have those things discussed. So I think the more we bring up these even minor features, the more likely they are to get the attention they need. So also important for our trial readiness. I also just want to talk about some other ways that we can try to predict, not only based on the 2-dimensional structure, but Dennis Lal and colleagues kindly went through the variants that we saw, the pathogenic and likely pathogenic variants we saw in the patients we looked at. And he plotted first the patient's variants in the red, and then a large group of individuals with variants in KCNQ2 from the UK Biobank who presumably didn't have epilepsy or any kind of DEE. And you can see there's a real split there. You can see that in the pore of the channel, you can see there's a clustering of these variants. You see that a little bit in the 2-dimensional structure, but you can see it more clearly in Dennis's nice images here. And it really does separate out. We then wanted to say, well, can we then figure out of the kids who have movement disorder or not, or early seizure onset or not, can we do the same thing? So here in this part, we've got the children with a movement disorder, this is ours as well as in the literature plotted in the magenta, and then in the aqua is everybody else. It doesn't really make a nice split. It's hard to predict because you have variance throughout, and it's hard to predict there. So I wouldn't say, at least with the numbers we have, we've got the tightest genotype-phenotype correlation there. Likewise with seizure onset, maybe a few more of the kids with later onset have variants up in this part of the KCNQ2 protein, but by and large, the others are sort of mixed through the protein. So we were struggling a little bit to find tight correlations there, but I think it's worth just trying to look in different ways for how we can predict. So this is a little bit more of Anne Berg's nice study that she allowed Sanal Mohita and I to participate in. We looked through at all the variants, and we vetted the variants of many of the participants in her natural history online interview study, and she looked at many different aspects that were non-seizure aspects, but she tried to relate them to the seizures, as John mentioned. So mobility, for one of them, there's a mobility score that really did not seem to depend on how recently your last seizure was. So this is looking at if you had a last seizure within the last 6 months, within 6 to 24 months, and then if your last seizure was 2 or more years ago. So no real connection there. With communication, there was a little bit of an emergence of a trend. It looked like maybe the kids with more remote seizures had more effective communication reported. That's a small number, though, but you did see children who had rare communication and minimal communication in all 3 groups, meaning those who had seizures in the last 6 months, those who had seizures over 2 years ago, and everybody in between. So an emerging trend, but it still didn't give absolute correlation. She also interestingly looked at something that, again, we don't always, as neurologists, think to focus on, but is really important from the life of a family, which is, can the children eat independently? And looked again there as sort of whether the recent seizures were a marker of severity. And there were a few children who were able to eat independently, and they did have seizures that were longer ago. That may just reflect that they were older, and that these are kids who had seizures earlier on. But I think what's clear is that there's a trend towards, the longer ago your seizures were, the less impacted you are by these other domains, which is a little bit hopeful in some ways. But as you can see, it's complicated, and there's lots of each of these different colors, and each of these represents a different group of kids with an intersection between their skill level and their seizure recency. So lots of different factors and not an absolute prediction. But again, things that are important, and the hope here is that if we focus on seizures early on and we get the diagnoses early and we focus on seizures early on, that it will actually impact these other domains, and we'll have to track that and see if that bears out. So some additional observations from the registry I wanted to point out. We've already heard earlier that we have reported this ezogabine experience. We're in the process of hopefully getting this formally published. We presented it in 2017, so it's kind of old news. But I did want to emphasize that out of 8 children, we had 5 responded with reduction of seizures from ezogabine. All of them, there was a report. Someone asked earlier about what about the developmental response to ezogabine or to the newer compounds that are like ezogabine, the channel openers. We had a subjective report of improved alertness or emergence of, I put milestones in questions. They probably were more inch stones, but clearly, when we asked again and again, what were they like before? What do they do now? And the improved alertness was things like, well, before when I walked into the room, they didn't necessarily seem to notice, and now when I walk into the room, my child looks over at me and coos and makes a noise, or when their sibling is now in front of them, they laugh, and they clearly are aware of the presence of their sibling. And so these are things that you've heard Anne Berg say many times, and others in the community. These aren't measured by our normal developmental scales, but these are the sort of inch stones that do make a difference when a child will smile and laugh when there's brother and sisters in the room and interact. So we did see that with ezogabine. I hope we see it with the newer versions of the channel openers. Unfortunately, 3 did have urinary retention, at least my own patient was in the local emergency department dealing with this, but stayed on the drug because of the improved alertness effect. When we had to take it away when ezogabine was removed, 3 had withdrawal seizures. A couple of them had pretty substantial irritability and regression, and one of them were in the process of trying to obtain trial, I should say, for that. So what else did we see? This is something new. This is something Brandon's written up, and you'll see it in a poster if you come to the AES meeting, and it's something that people in the community brought to our attention. Almost simultaneously, we heard from one family in the States and one family abroad who brought this hyperkinetic movement disorder and gait abnormality after exposure to medications that were used for behavioral symptoms. So I'm just going to go through in a little bit of detail what was reported, and to say that this is early, and this is what we like to call hypothesis-generating data that we get from a registry. So this isn't because we specifically asked. Now we have to go back and ask, but we didn't specifically say, hey, were you on this drug? Was your child on this drug, or this drug? These were spontaneous observations that families made to us and said, hey, we're seeing this in our child with KCNQ2 encephalopathy. Have you seen this before in the registry? And because two families came to us with these symptoms, and because our research team was so used to the sort of routine KCNQ2 enrollment, we heard about the first one. This was a young man who was put on paroxetine, which you may know as Paxil. It's traditionally known as an SSRI, or selective serotonin reuptake inhibitor, but it also has dopaminergic activity. It actually increases dopamine function. And this young man had a severe deterioration of his gait and also had some hyperkinetic repetitive movements that were sort of hard to classify. I heard about it directly because, as opposed to most of the information which gets collected and we catalog in groups when we have a large group to aggregate, the story was so unusual to Devin Knight, who was our research assistant at the time, that she said, Anne, I've just heard something I've never heard before from a family with KCNQ2. I think you need to call this mom. And I said, wow, Devin's enrolled like 200 patients, and she's never called up that night and said, I think you need to call this mom. So I called this lovely mom who told me the story. I said, yeah, that's unusual. It probably is an effect of the paroxetine. I had to look up. I'm a practicing neurologist. I trained formally in psychiatry. I think of paroxetine as a serotonin agent, but I had to look up and find that, yeah, there is a dopaminergic cross-reactivity. I said, that's interesting. It could be a dopamine effect. Let us know what happens when he comes off. And he did come off, and it was restored to normal gait and stopped having these abnormal movements. So I don't think it was just he had a movement disorder that suddenly was noticed. It really seemed to be a drug effect. We then heard from another family that there was another child who was a young woman who had had a previous exposure to Risperdal, which is traditionally thought of as a dopaminergic agent, but also has some serotonin effect. And then subsequently to aripiprazole, which has dopaminergic, predominantly the D2 subunit agonist, so it increases activity there. These are often very useful medications for behavioral symptoms, for irritability, for sometimes even for sleep, but they do have potential side effects. And tardive dyskinesia, meaning typically a late developing movement disorder, can be one of them. But we're taught in our training that it only takes one exposure, even one dose, to potentially have movement disorders result from these drugs, which can sometimes be long-lasting. So in this case, after much discussion, and actually much discussion in part because there was some efficacy for these medications, the drugs were taken away, and the symptoms improved pretty dramatically. So I bring this up because these are important stories that generate hypotheses to us that make us think, well, these could just be these children had these effects to these meds because these meds can do that. But when we've heard about a third individual actually where we're trying desperately to make contact and haven't been able to make formal contact, so I can't give you any details there. But the question really is this just a coincidence? These children received these drugs and they had these adverse effects, or is it somehow related to KCNQ2? We know KCNQ2 is expressed in the basal ganglia, which is the movement control center, very simplistically. Are you taking an already stressed system and then imposing upon it this dopaminergic change or this serotonergic change and bringing out symptoms that you otherwise might not see in a similar child who wouldn't have KCNQ2? We really don't know. We have to think about ways to potentially model this. But there's some things that we could do to try to answer that question. We can go back. So the beauty of having a registry is we can go back and say, how many exposures are there? So we know about these few exposures that resulted in these side effects. We don't know what the denominator is. So how many children were exposed to risperidone? How many kids were exposed to the SSRIs that have dopamine cross-reactivity? So this is something we'll probably be coming back and asking you, when we went through all the meds for seizures and we went through all the meds for, other medical concerns, we didn't specifically say, was your child treated for sleep or behavior? So I think we need to do that. So I think resurveying everybody or surveying to see if these meds had good or bad effects will be important. The other thing is, well, what about the other way? Looking at this from completely the other end. So lots and lots and lots of people are exposed to this category of medication for all sorts of indications, psychosis, various other symptoms, and so on. And our psychiatry colleagues are beginning to catalog those patients who are treated with them, looking at their effects. And so what if we looked at the psychiatry registries and if we could find a large group of individuals exposed to these patients and a subset who then had these side effects and look for things like KCNQ2 variants. It's a bit of a outside the box idea, but why wouldn't we do that? And I think as databases grow and are being collected for all sorts of reasons, our psychiatry colleagues might be able to help inform us and share their genomic data with us so we can take a look. So that's something that I hope we can do in a small level at our institution, where we can look across cohorts for different variants. We've already done that with some of our psychiatry colleagues with other genes that are DEE genes, but they're also relevant to their conditions, and something we could think about doing together longer term. So I wanted to sort of bring things together and talk about purple stars aside. Are we really ready for a precision trial for KCNQ2? Because we know things are emerging, right? We, again, didn't do a prospective assessment of seizures, development, movement disorder, medication, sleep, and all of those things prospectively over time for everybody, but we have been able to stitch together a set of patterns, right? We know that the seizures tend to start from the patient symptom perspectives. We have seizures, we've got a sense of what type of seizures, when they emerge, what they respond to, what they don't respond to, and so on. We have non-seizure outcomes that are getting cataloged, and we have quality of life measures that are being administered to families and can be administered. And if we can develop those for one rare epilepsy, I think we can apply them to many. So I think this is an area that's seen a lot of progress in the last few years, and that's going to serve not only KCNQ2, but many rare epilepsies. And then in terms of preclinical studies, the mechanisms of this disorder are pretty well understood compared to many others. And so I think we're in a good place there. Variants can be studied in vitro, and as Al showed you, many of them have been. And if they haven't been, Al and colleagues have lots of ways that they can do that. So I'm sure there's room for more assessment. And someone asked at the lunch, how do I know if my child's variant is pathogenic, if it's loss of function, gain of function? We can make some assumptions based on location and nearby variants, but if it's really in question, it might need some preclinical work to do that. But I think one thing we also have to ask ourselves is whether the preclinical models recapitulate the key core features of the disorder. And so there's the seizures and hyperexcitability, and then there's the developmental encephalopathy, which may be two different aspects we've got to think about. So I just wanted to talk a little bit about what else has emerged, which was really the direct result of the 2017 Peabody Summit. I had invited Casper Rowett, who is a postdoc with Clifford Wolf, my colleague downstairs, who studies sodium and potassium channels because of their relationship to pain. And speaking of sort of crossing fields, Casper was really interested in looking at some of the potassium channel variants with us. He went on to industry and brought Jaehoon Shim, his colleague, over from the lab. And Jaehoon's been working on, in a much smaller scale than Al's group, but he's been working on some iPSC-derived neuronal studies with us, and also collaborating with Maurizio Tagliatella's lab in Italy. And this was really, Jaehoon came and saw your kids and was so moved that he's devoted a huge chunk of his postdoctoral work to trying to understand what's going on in the dish. How does it relate to pain, and are some of the pain drugs going to be useful for epilepsy and vice versa? So that's, I think, a positive result you've all had on his studies, but certainly on the field. The first variant that we sought to look at is the S195 proline variant, serine to proline substitution. I'll be honest, it's because it was one of my patients and because we had access locally and we were able to collect a sample, and also I'll show you some of the results there. We also then took, here's a smaller list than Al showed you, but of several of the variants. We highlighted some of the recurrent variants and we said, well, let's start by looking at the recurrent variants. Al had published the data, or was about to publish the data on the threonine to methionine substitution. We said, well, if we can look at something where there's already traction in the field, where we know that there's in vitro assay potential for a phenotype, then at least we'll have that as a positive control one day. So we took it upon ourselves to do that. And when I say we, I don't mean my lab. My lab actually studies zebrafish models of candidate genes for epilepsy. This is the Translational Neuroscience Center that Mustafa Sahin runs, the translational research program that funded Jaehoon and Dr. Wolf's lab, and a number of different sources. And by having the registry and by having some samples in the freezer, we were able to sort of every time somebody says, hey, we're interested in potassium channels, we could say, oh, you want to study a few of the samples? We've got them in the freezer. And that's what led to all of these variants now being studied by our collaborators. And it sounds like a lot of people, but we all work within 2 floors of each other. And so it's sort of a pretty concentrated group of people who are studying these variants. So that's to be determined exactly what comes from all of these, but I'll share a little bit about the serine proline substitution. So a funny thing happened at AES a few years ago. We were standing at a poster, and Maurizio Tagliatella said, look, we've got a patient with a serine phenylalanine substitution, and it's loss of function, but I think your serine 195 proline substitution is gain of function. So I said, oh, that's unfortunate. We've put them on ezogabine. I hope it's not going to cause a disaster. And it didn't actually cause a disaster, which is interesting. So we've been sort of having this friendly collaboration slash push me, pull you about, like, is it really gain of function, loss of function, or what are we talking about here? So I'm not going to go into the details. You can ask Al exactly how to interpret all of Maurizio's data on this side, but I will tell you that just as Al did show you, there are ways to look at wild type and to look at different variants. And what I hope I can show you is that the triangles over here are to the right, and the triangles over here to the left. The 2 different variants had different effects on the membrane potential in these 2 different variant situations. So that suggested gain of function versus loss of function to Maurizio. With all due respect to Maurizio, my patient had seizures, the one with the supposed gain of function, and had done okay on ezogabine. It actually would have been one of the kids who had improved alertness pretty dramatically. I actually saw it for myself. So, well, that's interesting. He got better, thank goodness. And we had, we'd said we hadn't had in vitro studies before going on ezogabine, and we said if you get worse, we'll come off. But in fact, he got better. So Jaehoon has been doing what Al's been doing in his lab too, which is to take the cell lines that are made into neurons with this substitution, but then also doing some gene editing with CRISPR to make a corrected line. So we've got 2 sets of cells, and the only difference between them is this single amino acid at this 195 position. And hopefully these will play. This is what our patient's cell lines look like in a dish. These are, it's going a little funny. Let's see if I can get that to play again. Hold on. You see a certain amount of activity there. The lighting up is because we've got a fluorescent reporter there in these cells. And this is a control set where there are some coordinated bursts, but the corrected line doesn't seem to have as much of the activity. And just to make the point, the computer is making the patient's cell line go completely wild with activity. This is just some electrical glitch. But I think what hopefully will be clear is that we can get these cells to grow, we can look at the activity, and then what he's been able to do is to look and to quantify the activity between the patient cell lines that are turned into neurons and the controls. And in fact, there are, depending on how you put all the data together, there is some increased activity, and so it looks like there's hyperexcitability in the dish. What does that mean about the gain or loss of function? I don't know. So this is something we have to figure out because it really does look like with the traditional assays, it's having a gain of function effect by those parameters. But when we just look at very, very bird's eye view, how much are the cells firing? It looks like they're firing more than the corrected version. So that's not fully explained, but also something we can use as an assay if we're going to then screen with drugs. We can look at this fluorescence as one readout. This is a somewhat more complicated way of showing that we're looking at these firing rates and so on, and that when we have the corrected versions, the activity does go down. And I'm not going to go into great detail, but I just wanted to point out the red with all the spikes, lots and lots of spikes, and these red lines here represent the patient, and then the black is the corrected line with decreased amplitude, decreased response to various currents, and so on. And so we are seeing a difference. We hope that this is something that we can screen with medications. Proof of principle, let's look at ezogabine or retigabine as it's called in the lab, and did in fact suppress some of the activity in the abnormal line, so that was useful to sort of show we were on the right track. Jaehoon then used another compound, this XE991, which is a selective potassium channel blocker to show that if you block that, then you block the effect of the ezogabine. So it really is a specific effect, not just a random effect that other drugs might have. So we've got a system that works. We're now going to see if we can do some screening. And what are we going to use to screen? We're going to use all of the sodium and potassium channel and other ion channel affecting compounds that the lab has been using to study pain. So we're hoping this cross-fertilization will be effective. So in the last couple of minutes, I just wanted to mention we have started doing some drug screening with the Wolf Lab around SCN8A, and this is where I think the sodium and potassium channel work that they're doing is going to be helpful. This project just got a little bit more of a kickstart, but we have patient cell lines and corrected cell lines and see very similar patterns. So red over black, abnormal over normal, looks a lot like our KCNQ2 scenario. So I think we've got parallel systems working in similar ways where we have the ability to measure these things, and we'll hopefully be able to use this to do some drug screening. So they've been doing this now for SCN8A, and some compounds are emerging. I'm not going to get into all of the particulars, but this is just to show that you can plate cells, lots of cells at a time, expose them to, excuse me, hundreds of different compounds and see which ones of them decrease activity, increase activity, and then once you have some candidate molecules, maybe you can go after those and look at them in more detail. So hopefully more to come, and hopefully it won't be another 5 years before we have something to speak of there. So the last couple minutes, the emerging projects that we have going on, you heard about the in vitae studies, you've heard about Beyond the Seizure, which does free sequencing. We have a study that's connecting with the UK, Australia, and Canada called IPCHIP. It's an international pediatric child health consortium, starting with 4 hospitals, but hoping that will grow. We got our hospital leadership to invest in trio genome sequencing for infants with seizures. So we're diagnosing these kids on average within 3 weeks, but if we have a positive finding, usually within a week. It's a little bit of a pie in the sky model. We know this can't be done everywhere, but as genome costs go down, we think it actually can be done at least in the NICU. And so our colleagues in the NICU have been launching some community-based projects, and we're hoping to get this to more than just 4 centers. So we're going to find more children with KCNQ2 is the bottom line. They're out there, and we're going to find them earlier, and hopefully we'll have trials for them. I also wanted to point out, as John was saying, more epileptologists are sequencing. They'd better be sequencing, because we've had evidence for a long time about how important this is. We've had evidence for a long time about how important KCNQ2 is on the scene of newborn epilepsy. We've been saying it. We did a systematic evidence review with Beth Scheidley leading that last year, which was published in Epilepsia, and Lacey Smith in our group worked with the genetic counseling organization to have guidelines that are just published this month and formally endorsed now by the American Epilepsy Society. So nobody should ever be told that it's not worth sequencing an unknown epilepsy, whether they're a neonate or they're in their 20s or 30s, and you now have published guidelines to back you up. Sarah's going to be hopefully talking about this, but I know Sarah's launching a whole genome sequencing modifier study, which we were hoping to piggyback on. And Aubrey's here and can talk to you in the break a little bit about a study for looking at what we call haplotypes. I'm going to try to use her slides here to say that this is not at all what we've been talking about, but we all have what are called haplotypes. There's just groups of genetic variations. We all have little variations in our genes. Some of those are going to be near KCNQ2 and some of them are not. And if we can find variants, little genetic changes that we have or that your children have near KCNQ2, it might give us a little bit of a hint as to how to target treatments. So if somebody has a KCNQ2 variant, let's say here, and they've got these little variants, we call them SNP or single nucleotide polymorphisms, right near KCNQ2, we could target those. And the nice thing there would be that you wouldn't just be able to treat this child, but you'd be able to treat any child with the same array of SNPs. And so this is an attempt to find out what's in the vicinity of KCNQ2 and find groups of children, regardless of what their specific variant is, but groups of kids who might be lumped together and grouped together in future trials with a number of different strategies. So hopefully by doing that, we can take genome data. Aubrey, Tim Yu, and others in Tim's lab will hopefully generate some sort of molecules, such as ASOs you've heard about, to target them, and then we'll be able to have not just one child, but a whole group of children who might be able to benefit from that. So this is really at the chalkboard stage. Someone was asking about a timeline. I'm not going to give you a timeline. It's very much at the early developmental stage. But people have asked what you can do to participate, and I'll tell you what you can do to participate and what we need, and you can decide if it's appealing or not. If you're interested in updating your registry data, we will ask you for some time, and we won't do that here at the conference, but we'll be doing that over the next few months to try to get some of the details of what we missed the first time around that we didn't know to ask. If you haven't enrolled and you'd like to enroll in the registry, there's the consent process and the time, and I really hope we can link with Citizen and other groups so we don't have to take as much time as we took from the families who started this with us and have already given us hours and hours. If you'd like to participate in the genetic studies, as with all of these, it's completely voluntary, but Brandon Obi, who's here, is happy to do consenting. We thought we would have you find us after the session between now and the dinner, between the break and the dinner, sign up with Brandon. He'll set up a remote consent time. We'll send you tubes. We can send them to Australia. We can send them to wherever. And this would require blood samples. So we can not only look for modifiers for Sarah, but also look for the SNPs for Tim's lab and others' labs to sort of say, let's guide you all in terms of how you can help the KCNQ2 community. So I hope that'll really help us moving forward. This is very chalkboard. So this is going to be, Jim's going to say, where are the results? And I'm going to say, Jim, we don't have them yet, but you should ask me every year, because then I can go back and say, hey, they're waiting. So with that, I'll just thank all of you for your attention. Brandon and Sanal for your very hard work on our registry, but also keeping everything together. And Al, Sarah, and Sarah for putting that lovely element together where all of these and more can be summarized. Thanks so much.
Questions? Anyone? Any questions? You know what? I think if we have questions, let's do them kind of as, Brandon, can you stand up so if people want to get consent or sign up for anything, Brandon is the person, and Anne, who you see. All right, well, we're going to move on, and thank you so much, Anne Paduri. We really appreciate it.